Everyone. I'm Dr. Aisha Siddekha from Government First Grade College, Tumkuru. To be honest, like after listening to all these beautiful and wonderful research work that is being carried out by all the presenters, I'm really feeling very inadequate because we come from a very humble background, and since I don't, this is not my original research. I would like to clarify on that one. I'm just trying to give overview. In fact, when I asked the organizers, they said we can present a review. But honestly, I'm feeling like the wonderful work is being carried out by many of the presenters. I really congratulate all of them. I'm just going to rush through what I have summarized about. My topic is nanomaterials in optoelectronics. So I really feel very inadequate. Kindly excuse me for this one. So what I thought was like I'll talk about how the nanomaterials are being helpful in various aspects of optoelectronics, including energy conservation and conversion, because right now we are going through a lot of energy crisis. So it becomes essential with the dwindling natural resources and the fossil fuels. We are facing a lot of problems even in terms of pollution, and everybody is working on generation of green energy, and keeping the environmental safety is a major concern for every one of us. But at the same time, the requirement for energy conservation and storage is becoming very essential because everyone is in need of this one, including the industrial, domestic as well as service sector. So it becomes essential that energy conservation is a very important aspect for us right now. So in this aspect, how the nanomaterials are helping us is what I thought of highlighting. So nanomaterials are being used in various places such as LEDs, OLEDs in various forms like nanocomposites, nanowires, nanofluids, sheets, and nanorods. And we are using various types of nanomaterials, whether they're directly metal nanoparticles or metal oxide nanoparticles, metal titanates, and some of them are magnetic, as well as we are also using carbon nanoparticles. So when it comes to nanoparticle applications, these applications we find use in sensors, as we are listening to so many people here, energy storage, energy harvesting, and usually then these nanomaterials, as everyone knows, are being prepared by all these methods which I have mentioned here. So I thought I'll just tell a few things about this. Nano lasers, they're very powerful, they're compact and energy efficient compared to the conventional lasers, and they're being used in various places such as sensing, spectroscopy, especially in biological probing, probing of cells, DNA, etc., as well as gas detection. And we also have the lithium ion batteries, which are useful for energy storage. But now with the nano LIBs, there is a better rate of both charging and discharging of the batteries, and they're very useful in the portable applications such as mobiles and laptops, etc., due to their increased conductivity, thermal regulation, and other things. And we also have the nano LEDs, which are responsible for all those beautiful things that we are seeing and watching nowadays in the form of displays, and they offer better functionalities. And finally, the are being also used in ultra capacitors. These exhibit higher capacitance compared to the conventional capacitors. They will be once again very useful in portable devices such as hybrid vehicles, mobile phones, etc. Coming to the types, this is what I thought I can summarize. We are using many types of nanomaterials, whether they are like metal oxide, in which we can use zinc oxide, copper oxide, calcium oxide, magnesium oxide, various things which have been combined with other materials like polymers, etc., and other organic materials to form the thin films or rods or whatever. And finally, the thing is like the use is dependent on the way we fabricate it, and we can design materials as per our requirement. Then continued part, I've just tried to say a few more things like fabricated blend of nanocomposite using PVP and carboxymethyl cellulose. This is very useful in radiation shielding applications and something like smart windows and etc. So some of these are also being used as humidity sensors, just as we saw now. And iron oxide nanoparticles, they are useful in charge transfer and many other functionalities. One thing I just wanted to draw the attention is like nanomagnetic suspensions, which are known as magnetic ferrofluids. They're also very useful in various applications such as magnetic resonance imaging, drug targeting, etc. Next, similarly, gold and silver nanomaterials are also being used for almost all these kind of purposes. So finally, the carbon nanomaterials, whether it is one-dimensional, two-dimensional, three-dimensional nanomaterials, they also possess unique optical and electronic properties. They're also being used for various purposes in optoelectronics. And metal titanates in the form of zinc, calcium, and even barium, they all find use in various places in UV optoelectronic devices, energy harvesting, holography, etc. So this is just a summary. Like now, nanomaterials are contributing hugely to the electronics and our well-being, and we're all progressing digitally because of nanotechnology. And a huge amount of research is being carried out in terms of synthesis, characterization, as well as the applications of these nanomaterials. So this is what I just wanted to conclude, and thank you.
So first of all, I am thankful to the organizing committee to give me opportunity to present my work on this conference. I am Dr. Kumar Gaurav, working as an assistant professor in Namath University, Haryana, and my field of specialization in different type of the biophase like the biotin cell, bioethanol, and the microbial fuel cell. So in this process, we are using the microbial fuel cell to produce the electricity. So this is the sustainable energy solution. So first of all, we will discuss about what is the microbial fuel cell. So microbial fuel cell, that is the electrochemical device, or we can call it as the bioreactor. So in that one, different type of the process is going on with the help of the microemulsion, so that this chemical energy which are stored in different type of the organic matter, which is converted into the electrical energy. So this is the cause as a renewable source of the energy, because in that one we are using different type of the materials. So it may be the organic matter, different type of the waste, different type of the sludges. So this is called as the renewable source of the energy. So in general process, the microbial filtration, that is consist of the anaerobic chamber, that is the anode, aerobic chamber, cathode, and in that one connecting both aerobic and the anaerobic chamber with the help of the proton exchange membrane. So for that ones we can use the different type of the exchange membrane like the Ultex, Nexion, different type of the salt. So for that ones, we are not prefer the Ultex membrane, Nexion membrane, because these are the costly membrane. So in place of that ones, we have synthesized the different type of the membrane, and they provided the good type of the electrical conductivity. So now we'll discuss about what is the schematic diagram of the microbial filter. So in that one, the left hand side, that is the anodic chamber, right hand side, that is the cathodic chamber, and the both the chamber which are connected with the help of the proton exchange membrane, that or we can call as the salt bead. So in the anodic chamber, so in that one we inoculated the microbial like microbial cells, and in that one we provided the substrate. For example, if we provided the glucose, so glucose in that one after the conversion, so it produces the carbon dioxide, produces the 24 hydrogen and 24 the electron. So this 24 electron can be transferred into the anode, and this can be transferred into the electrical circuit, and the proton, around the 24 protons which are transferred to this proton exchange membrane. So in that one, it produces the electricity. So in this one, we can use the microbial cells, the microbial fuel. So in that one, microbial cell, it may be the bacteria, any type of the other type of the microorganism. So this is the microbial setup. In that one, we have designed in the left, in that one, the anodic chamber, cathodic chamber, and we have used the, with the help of the multimeter, we can use to measure the voltage like in that one. So first of all, we can take the microbial culture like the bacteria Alphutin faecalis. In that one, we procure from the National Chemical Laboratory NCIM. So in that one, this was the culture was maintained in the LB plot. Out of that one, we have taken the anodic material, that is made up of the graphite rod, and the cathode we have used the copper plate. And before using that ones, they need to sterilize. So for that ones, we can use the absolute alcohol. After that ones, we have aerated with the UV addition for the 30 minutes. And for measuring of the voltage and the current, we have used the digital multimeter. So for that one, we have can check the what is the efficacy of the membrane or what is the efficacy of the microbial field performance. So that ones we have calculated the power density, that is the VI divided by A. And A is the area of the electrode, and V is the observed voltage, and V is the observed current. After that ones, we have synthesized the membrane. So in that ones, we have uses the dimethyl formaldehyde 50 ml, and in that ones we have added the pharma, that is the polymethyl methylicate, and pellet was the dissolved in the mixture and stir for the 40 minutes. After that ones we have added the dopamine. So dopamine it can increase the conductivity. So for that ones we have used the potassium thiocyanate and the citric acid. So after that ones we have can changing the concentration of the potassium thiocyanate and the citric acid. We have the different type of the membrane. But in this presentation I have shown the one type of the combinations. Yeah. So in that ones we have used the addition citric acid and the sodium potassium thiocyanate, that is the 2.5 ml. After that ones we have resulted in the solution in the effective plate. After that ones we have heated this membrane in the hot oil oven 60 degrees for the 6 hours in order to remove different type of the solvent, and it can be prepared within the 2 days. All that ones we have characterized what is the property of the mineral. So for that ones we go for the SEM analysis. So analysis which can provide the surface morphology of the prepared membrane. After that one we have can check the what is the quality, or we can check what is the conductivity of the membrane. So for that ones we have checked the water uptake rate. So water uptake in that ones we have take the dye sample and immerse in the 24 hours in the container which have the distilled water, and we kept at the room temperature. After that one, the surface was removed with the absorbent paper and to get the dry weight. And we can measure the water uptake. So in that one, water uptake, that is a W minus WD divided by WD, and then multiplied by the 100. After that ones, we have checked the ion exchange capacity. So this in that ones we have how to calculate this one. So we go for the classical method for the ion exchange capacity. So what we use, we have used go for the titration method. So in that ones we have used the 1 molar solution and for 24 hours, and then 20 protonic. After that ones, this was washed with the large amount of the water to remove the excess acid, and we go for the titration with the help of the 0.1 molar NOH, and annexing capacity was calculated with the formula, that is the milliequivalent per gram of weight and multiplied by the volume of the NOH. So now we come into the discussion part. So first of all, we go for the surface morphology. So surface morphology of these days, no aggregation of the dopant like the potassium thiocyanate and the citric acid. So this is the uniformly distributed, and the presence of the salt can be clearly seen, that is the surface of the membrane can be doped with the salt. So this is the SM analysis. After that one, we have compared with the virgin membrane and the synthesized membrane. So for the synthesized membrane, that is the water uptake, that is around 4.1%, but in case of virgin mix, in that one there is no addition of the dopant. And after that one, we have calculated the ion exchange capacity, so it can be assessed to the proton conductivity of the membrane. So ion exchange capacity of the doped membrane, that is the 0.24, and the virgin membrane, that is the 0.1 equivalent. And we have calculated the power density of the synthesized membrane. So power density was the 22.4 milliwatt per centimeter square. So now come into the conclusion. So in that one, the microbial field cell which can be exploding the water. This one which can be used as a fuel cell for generation of the electricity. So integration of different type of the dope carried out the potential to realize this system, enhancing their efficacy, scalability, and the sustainability. And in that ones, we are also trying to use different types of the photon exchange to study the different type of the wastewater treatment. So we can use this wastewater treatment as well as the electricity generation. So this is the dual process for the renewable source of the energy. So this is the thank you.
From Government Navin College, Saragaon, Jahangir Champa, Chhattisgarh. The topic of my presentation today's work is the pressure and ionic conductivity measurements of a new sodium ion conducting blended solid polymer electrolytes. And now the development of new polymer electrolyte is one of the important areas of research in last 25 years. And these are the potentially used to develop solid state devices like batteries, sensors, and fuel cells. Is slide is moving or not, sir?
Sir, we are on the first slide. Can you please try again?
You can sit with her and you can discuss whatever. Please. ठीक है बच्चा?
I think slide is not moving.
It is not changing, sir. Please try again.
Once again, I am trying.
Your cursor is moving. Can you click on some slides?
Yeah, I am trying to move, but it is not moving, I think.
Sir, can you please press the F5 or the entire screen presentation mode?
Yeah, it is moving, sir.
Now it is moving. Yes.
Yeah. Now the polymer electrolyte shows many advantage, and this is the lightweight, flexible, high ionic conductivity, high mechanical strength, and good electrolyte compatibility. These are the some polymeric based batteries available in the market at different shapes and sizes. Now in present investigation, we have synthesized a new sodium ion conducting blended solid polymer electrolytes 70 PO, 30 NaCl and PVP. And characterization, their ion transport properties like conductivity sigma, ionic mobility mu, mobile ion concentration n, and ionic transference number tin. The advantage of the sodium ion salts over the other alkali salt is it is relatively cheaper, abundantly available, less prone to miser, and less reactive, and this can be easily handled in open ambient conditions. Now in experimental, we have firstly synthesized the blended solid polymer electrolyte. These are the starting chemicals, PO, NaCl, and PVP, taking at a different weight ratios, then mixing in a crucible, then again mixing in heated steel blocks, and that pressed using this caviar pressure machine, and we found a polymeric film with the approximate thickness of 0.016 centimeter. Now in ion transport studies, we have calculated their ionic conductivity. Sigma is equal to L upon Ra, where L is the thickness of the sample, R is the bulk resistance obtained from the LCR bridge, and A is the cross-section area. Ionic mobility and mobile ion concentration we have also calculated by the transient ionic current technique using the XYT recorder. With the help of this formula, mu is equal to D square upon V tau, where D is the thickness of the film, V is the applied potential, and tau is the time of flight obtained from the XYT recorder. And we have also calculated the mobile ion concentration using this formula. N is equal to sigma upon Q mu. Now in result, this is the compositional dependent conductivity of PO and NaCl. We can see here as the concentration of sodium chloride is increases, conductivity is also increases, and the maximum conductivity is obtained at 38% of sodium chloride, with the ionic conductivity is 4.6 into 10 to the power minus 7 Simon per centimeter. This has been used as a first phase host matrix for the formation of present blended solid polymer electrolytes. This figure shows the compositional dependence study of blended solid polymer electrolytes. This is the host salt, and this is the another polymer, polyvinyl pyrrolidine (PVP). And we can see here as the concentration is increased at the 2 weight percentage of PVP, their conductivity is So the maxima with the ionic conductivity 3.7 into 10 to the power minus 5 Simon per centimeter, and their one order conductivity enhancement were observed at this particular composition, and this composition is known as optimum conducting composition. And further increasing the concentration of PVP, their conductivity is decreases. The decrease in conductivity is due to the blocking effect created by the polyvinyl peroxide in this blended the solid polymer electrolyte systems. Now this figure shows why the conductivity is increases. What is the reason behind the conductivity increases? We can see here, this shows the ionic mobility and mobile ion concentration plot. And as the concentration is increases, ionic mobility and mobile ion concentration both are increases, and the maxima is found at the same weight percent. It means the overall increase in conductivity is due to the increase in both ionic mobility and mobile ion concentration. These are two factors are responsible for the conductivity enhancement in the present blended solid polymer electrolyte systems. This figure shows the exotic patterns of pure PO, SP host, and BSP OCC. As this star shows the blending, this is the source, the presence of PVP. Yes, we have blended the host polymer with the PVP. At the 20-degree, its peak is appearing here. It means it is the complexation of the polymer with the PVP. Now this figure shows the surface morphology of blended solid polymer electrolyte optimum conducting composition. This shows the smooth surface morphology, but while the pure PO shows the very roughness and a rough type of surface morphology. The second figure shows the differential scattering calorimetries, and this endothermic peaks shows their melting point temperature. This is the melting point temperature of pure PO, approximate 70 degrees Celsius. And in this blended solid polymer electrolyte, their melting point temperature is shifted towards the lower side. It means their flexibility is also increases, so their conductivity is also increases. And this is the great evidence for the conductivity enhancement of our blended solid polymer electrolytes. This table shows the comparative studies of the different ionic parameters. This is the conductivity of pure PO, polyethylene oxide, and this is the conductivity, mobility, mobile ion concentration, ionic transference number, and activation energy of this SP host. And this is the ionic conductivity, mobile ion concentration, and other parameters of the newly synthesized blended solid polymer electrolyte. We can see here clearly their conductivity increases up to the one order, and this increase in conductivity is due to the increase in both ionic mobility and mobile ion concentration, as well as their activation energy is also lower as compared to the SP host salt. Now this figure shows the temperature dependent conductivity studies of SP host and the BSP OCC. We can see here as the temperature is increases, conductivity is also increases, and at this particular temperature, their jump in conductivity is observed. The jump in conductivity is due to their melting point temperatures. And below this melting point temperature, if we calculated the activation energy by using the Arrhenius equation, and we have found activation energy is 0.37 in case of SP host and 0.21 in case of PSP OCC. It means the conductivity decreases, and this decrease in conductivity or lower activation energy indicates that this can be potentially used for solid state device fabrications. Now, this figure shows the ionic transference number measurement. It means how much percentage of ions are mobile in these systems. With the help of this formula 1 minus I upon IT, and with the help of this graph, we have calculated the ionic transference number, and we have found T ion is a 0.95. It means 95% of sodium ions do take part in the conduction process, and these are the responsible for conductivity enhancement in the blended solid polymer electrolytes. Now, finally, we have concluded a new sodium ion conducting blended solid polymer electrolyte. This composition with ionic conductivity 3.7 into 10 to the power minus 5 Simon per centimeter have been synthesized by a recently developed novel hot press solvent-free technique. And this technique we have um, invented in India first time at 2009, adopted in our research laboratory. And the ionic mobility and mobile ion concentration study of blended solid polymer electrolyte shows the overall increase in conductivity is due to the increase in both ionic mobility mu and mobile ion concentration. These two ionic parameters are responsible for the overall increase in conductivity. The lower activation energy 0.21 electron volt is in blended solid polymer electrolyte is indicative of it is the easier. This is the not silver. This is the sodium ion migration in the system. There is the typing mistake, and this is the Na plus ion migration in the system. And the ionic transference number measurement indicates that the 95 of total available sodium ions do take part in the conduction process in the newly synthesized blended solid polymer electrolyte system, and this can be potentially used for solid state device fabrication. Especially we are working in polymer fabrication of polymer batteries. Thank you very much.
Yeah, so thank you very much, and a very good afternoon to all of you. I am Praveen Dahiya, professor from Center for Biotechnology and Biochemical Engineering, Amity University, Uttar Pradesh. I'm going to present on the topic green synthesis of iron alginate nanogels by microemulsion method and Taguchi-assisted encapsulation of Candida rugosa lipase for the synthesis of butyl butyrate ester. So to begin with, we need to know what are these esters and how important they are. So esters are fragrant organic compounds used as flavoring and aromatic agents in industries. These are required in highest volume of industrial organic compounds, which are produced with a global market size of 89 billion US dollar at present, which is expected to reach $135 billion by 2032. These are used as flavoring and aromatic agents for the production of jellies, chocolate, cakes, beverages, perfumes, etc. Now, how these esters, they are synthesized? So there are basically two different routes by which esters can be synthesized. One of them is the chemical route, which is mainly used in the industries. But the chemical route synthesis method is having several drawbacks. The drawbacks includes that the waste that is produced is hazardous in nature, and there will be several racemic mixtures with undesirable side products. So enzymatic transformations, they are employed for ester synthesis to overcome these challenges of chemical esterification reaction. Why enzymes? Because enzymes, they are effective biocatalysts under mild reaction condition that result in the production of high purity fragrant compounds. Now, this method where enzymes are used, it is going to follow the principle of green chemistry, and it aligns with the 12 principles of green chemistry, making them environmentally sustainable candidate for ester synthesis. So here, for the present work, Candida rugosa lipase, they are selected. Why they are selected? Because these enzymes, they are widely used for biotransformation reaction, as they have broad specificity, stability, as well as they have got the status of genetically recognized as safe. Now, enzymes, they can be used in the free form, but free lipases suffer several drawbacks. And in order to overcome these drawbacks, which the free lipase suffer, immobilization is an alternate. So this study is basically exploring the use of iron alginate nanoparticles as a support matrix for Candida rugosa lipase and its immobilization via microemulsion method. Now, alginate is used here because it's a biopolymer, and biopolymers, they are having several advantages, including enhanced enzymatic stability as well as larger surface area. Microemulsion method is comparatively an effective way to synthesize these nanogels, and here we have used a vegetable oil, that is virgin coconut oil, which is used as a stabilizing agent, and it is going to make the entire process more greener and safer. Moving on to the methodology. So previously, iron alginate nanogels, they were synthesized through water in oil microemulsion method, where organic solvent hexane was used. Now, in the present study, we have replaced the organic solvent hexane with a vegetable oil, that is virgin coconut oil, so as to have a green synthesis of iron alginate nanogel. Furthermore, the synthesized iron alginate uh, nanogel was morphologically as well as kinetically characterized, where several aspects were studied. This is then followed by optimization studies using Taguchi design and L16 orthogonal array, where immobilization parameters are optimized for Candida rugosa lipase synthesized using green technology. Effectiveness of this immobilized Candida rugosa lipase was studied for butyl butyrate ester synthesis, and this butyl butyrate is basically a pineapple flavor ester which has got several applications in food, cosmetic, pharmaceutical, and beverage industry. Response surface methodology and box Pencom designs are used for optimization of ester synthesis parameter, and esters synthesized, they are confirmed via FTIR and gas chromatography. Now, moving on to the results. Till now, hexane has been used as an organic phase, and it is due to its optical transparency. But here, in order to synthesize the iron alginate nanogel via greener approach, the hexane is removed from the system and it is replaced by virgin coconut oil. Rest all the methodology, that is water and oil reverse micelle, is the same. Only thing is that the hexane is replaced by the virgin coconut oil. So green synthesized iron alginate nanogels, they were developed using water and oil microemulsion method, which is further characterized via DLS, zeta potential, and scanning electron microscopy. The nanogels, they showed an average diameter of 100 nanometer and a zeta potential of minus 28 millivolts for immobilized Candida rugosa lipase, which indicated that they are more stable in the virgin coconut oil based system. Similarly, SEM images supported the elongated rod shape of the iron alginate nanogel with the size ranging from 80 to 90 nanometer. Morphological characterization thus confirmed the stability of these nanogels in the virgin coconut oil based system. Further, all the various kinetic characterization parameters, they were studied, where free and immobilized Candida rugosa lipase was compared, and it was observed that the immobilized lipase showed Km of 1.2 millimolar. It indicates it higher affinity towards the substrate. Similarly, it has also shown enhanced temperature, optimal pH, half-life, storage stability, as well as overall reusability up to 9 cycles. So based on the comparative analysis, it was observed that the immobilized lipase showed better properties when compared to the free enzyme. And further, Taguchi optimization of immobilization parameters were done. Taguchi is a method which is used for optimization of immobilization parameter, where Minitab 21.1 software was used for the optimization and validation, and L16 orthogonal array was used for the design of experiment. 4 different variables were selected, and these 4 variables includes temperature, substrate concentration, pH, and reaction time. Optimization using Taguchi method revealed optimum conditions at a substrate concentration of 0.7 microgram per ml, temperature of 53 degrees Celsius, pH of 8.5, and a reaction time of 20 minutes. The computed F value is 8.80, which implies that the linear model is significant at 95% confidence level. Out of the 4 important parameters, the temperature, pH, and time, they are showing that these are having greater impact on the immobilization of the lipase, whereas substrate concentration is having least impact. The enzyme activity, which was predicted, was 64.18, and where the actual enzyme activity is also almost the same, that is 62.11 micromole per minute. Optimization of the process parameters of butyl butyrate synthesis was done using response surface methodology and box Bankum design. The 3D plots are generated, and they are to check the effect of independent variables on the dependent one. Mutual interaction between temperature and time showed, it exhibited a strong and perfect interaction as shown by the sharp elliptical contour as compared to that of stirring speed and time, and followed by substrate molar concentration and time. The highest percentage yield, that is 90% of butyl butyrate, is perceived at the optimum conditions, where substrate molar concentration is 1:4 at 45 degrees Celsius, within 6 hours of incubation time. Further, the reusability of Candida rugosa lipase was checked, and it was observed that it is stable and it is showing highest conversion till 7 cycles, which contributes to the sustainability and economic viability of the product. Water activity was also determined, since it is a very important parameter when we talk about how synthetic and hydrolytic reactions are taking place, and a lower water activity of 0.3 was observed, which is giving significant esterification reaction. At the end, the esters produced, they are confirmed using FTIR and GC. This is the FTIR spectra for butanol, butyric acid, and butyl butyrate. And the FTIR spectra, they were compared with the standard FTIR spectra, which confirmed the production of butyl butyrate esters. The chromatogram for the samples, the GC chromatograms, they were even analyzed at 0 hours, which indicated that butyl butyrate was not produced till now, but hexane, butanol, and butyric acid, they are eluted at 1.5, 1.7, and 3.63 minutes, and butyl butyrate was seen at 1.905 time duration after 6 hours of the reaction. Such observations indicated that the ester is successfully producing in the presence of immobilized Candida rugosa lipase as a catalyst. Finally, the conclusion and the future perspective. So out of this work, we are able to develop an efficient support matrix, which is an iron alginate nanogel via greener synthesis, where Candida rugosa lipase is successfully immobilized, and this complete material is able to show significant efficiency in the synthesis of ester. Thus, it is now acting as an enzyme of choice for several industries, and it is even viability for commercialization. Developed biocatalysts can also be used for several other applications like biodiesel production, pharmaceuticals, and synthesis of other flavored esters. Now, this entire work aligns with the principle of green chemistry, providing to the growing demand for natural and environment-friendly products in various industries. With this, I would like to thank you.
Now, myself, Dr. Manjeet Singh, from Assistant Professor Zakir Hussain Delhi College, University of Delhi. And my topic is, as mentioned by the speaker, that is GIS-based multivariate analysis of the groundwater uranium in the central Ganga-Yamuna basin of the India. Our main aim is of this research is the research of groundwater uranium. But after a part of this, we have also analyzing the 18 parameters that are physiochemical parameters also. And these are the two monsoon seasons we have taken for this, that is the pre-monsoon season and the post-monsoon season. And for this, we also take 3 regions to collecting the samples of the water, that is the upper region, middle region, and the lower region. The sample analysis are characterized with the 2 various methods. First is the in situ methods, and second is the ex situ methods. Now some introduction regarding the uranium. Uranium is a heavy metal and is in the actinide series, and no stable isotopes, but also having 3 isotopes, that is 234 uranium, 235 uranium, and 238 uranium. Its level in the drinking water is less than 0.125 ppb, that is the permissible limit by the government. It is more chemically toxic than the radiological. Isotopes have similar chemical properties but different radiological properties. Uranium is a health hazard only if it is taken into the body and because it is an alpha emitter. External exposure is generally not a major concern because uranium emits only a small amount of low energy gamma radiations. The primary means of the exposures are ingestion of food by the means of the inhalation, or you can say the insignificant as the uranium air activity concentration is very low. Major health concern is kidney damage that is caused by the chemical toxicity of soluble uranium compounds. Uranium is a well-known nephrotoxic element. It is specific activity of uranium found in the nature as well as is compared to the neutral radionuclides. Therefore, the chemical toxicity superates the radiological effects. When the pH value is below the normal pH level, that is 7.3 in kidney, the dissolved uranyl carbonyl complexes decomposes in the acidic environment to uranyl and the bicarbonate ions, that is breakup of these ions. Uranyl ions depress the glow molecular functions because proteinuria and the albuminuria in the result. These are the our study area. So study area is located in the between the 26.74 to 28.49 north latitude, and this is the E longitude. Most of the area is agriculture dominated and heavy alluvial soil. Study area consists of the Bandar group, Banda alluvial, Old alluvium, Ajabgarh group, terrace, and the channel alluvium of the major river. This is the map, the land use or the land cover area of the map of the study. This is the LED fluorometer that is used for the testing of the uranium compounds. Before testing of the uranium compound, the sample is collected in the polyethylene sampling bottles, and that are used. The bottles are used after washing the 10% ammonia nitric acid, and then rinsed with the double distilled water, and then the sample is collected in the running water after 2 to 3 minutes, and then preserved with the nitric acid. These are the physiochemical parameters that we have tested in situ and ex situ method, that is the GIS coordinate by the GPS or GIS application, pH, TDS, temperature, dissolved oxygen, oxidation reduction potential, gamma radiation, total hardness by the ex situ, that is the titration method, calcium hardness is also titration, magnesium is also the titration. And these are some other parameters: uranium, chloride, total alkalinity, carbonate, bicarbonate, nitrate, sulfate, phosphate, and the fluoride. These are some results and discussion. This is the uranium contour plot. On the basis of this, we can find out the dark black part or the brown part is showing the maximum of the uranium part found in the water. This is the uranium that is found in the upper region, that is the pre-monsoon, and post-monsoon is 4.95 micrograms per liter, and in the post is 3.34. And in the middle, that is the 4.81, and pre-monsoon is 5.65. And in the lower region, this is 11.75, and post-monsoon is 14.04. That is the highest amount of uranium found in the post-monsoon. This is the Moran's method by which we can calculate the comparative studies of the spatial autocorrection of the things. This is the pH distribution of the upper, middle, and the lower monsoons. This is the Moran's autocorrelation of the pH, that is in the pre-monsoon and the post-monsoon in the upper, middle, and the lower region. The TDS is also found in the same pattern, that is maximum is found in the lower of the pre is 1061 milligrams per liter, and in the post monsoon this is 1178 milligrams per liter. The TDS that is in the Moran's method, this is the fluoride that is maximum is also found in the lower region of the pre monsoons and the post monsoon season. This is the fluoride. This is the gamma radiations. Gamma radiations, actually it is found in the upper region, that is the pre-monsoon, and that is the 140 NSV per H, and post-monsoon that is 135. This is the Gamma's Moran's method. These are the Pearson's correlation of the different parameters that we have taken in the upper, middle, and lower region for the both seasons, that is the pre-monsoon season and also the post-monsoon season. Here we have bold some parameters which are showing the highlights of this research. These are the some special correlation in accordance with the Moran's method, that is the TDS and the total alkalinity. These are the PCA graphs, that is the principal component analysis of the upper region in the pre-monsoon season and the post-monsoon season. And second one is the PCA of the middle region, that is in the both seasons, that is the pre-monsoon and the post-monsoon season. And this is the lower region of the PCA, that is the pre-monsoon and the post-monsoon season. The conclusion of our study that the uranium varied from 0.10 to 14.04 ppb. 5.45% of the groundwater sample exceeds the WHO permissible limit, that is the 30% ppb. 12.07% of the lower region groundwater sample exceeds the WHO limit. No significant difference were observed in the groundwater uranium concentration due to the monsoon precipitation. In higher TDS groundwater, uranium disturbed is clustered, and in the low TDS groundwater, uranium distribution is random. The calcium uranyl tricarbonate is dominating uranium species in the studied area from the groundwater. We acknowledge this from the BRNS and the BART, and also the chemistry department of the Delhi University and the Jefferson Delhi College. These are some publications regarding the same of this, and